Swayam Prabha. Digital India. Educated India.
Dear students, you are welcome in this class. I am Dr. Dinesh Kumar, Principal Scientist from Division of Agronomy, ICAR, Indian Agricultural Research Institute, Pusa Campus, New Delhi. This is lecture number 12 of the course Agronomy of Oilseed, Fiber and Sugar Crops. The topic of this 12th lecture is Insect Pest and Disease Management, aflatoxins and quality issues in groundnut. So, learning objectives are to describe the insect pests and diseases management options in groundnut crop. So, here you will learn the most important insect pests that infest the groundnut crop, and also you will see the diseases, their symptoms and their losses, and how to manage these insect pests and diseases in groundnut crop. Followed by you will also learn aflatoxin contamination. This is very serious issue in groundnut pods or groundnut seed, which has attracted attention of many people. And we will see how these aflatoxins entered the groundnut fruits or seeds and how to manage this problem. And also you will also know some seed oil and cake quality issues about the groundnut crop. But before going into the details of this talk, let us see some difficult word in the beginning. Arthropod, an animal with jointed appendages and an external skeleton such as crab, insect, mite or spider. Bacillus thuringiensis, normally called as BT, a group of bacteria that causes disease in certain insects. Formulations of several subspecies of Bacillus thuringiensis are used as insecticides, most commonly for caterpillars, but for other pests as well. So, these Bacillus thuringiensis is also used in biocontrol, and it is particularly important for organic farming, especially in groundnut. Biocontrol, the reduction of an organism's abundance or damage due to a natural enemy such as predator consuming prey. Biotic disease, an unhealthy condition caused by pathogen such as bacterium, fungus, phytoplasma or virus, means any disease which is being induced by a living organism is biotic disease. There may be some diseases which are kind of disorder. They are not caused by living organism. They may be caused by some stresses, nutrient stress, water stress, maybe soil pH, maybe certain climatic conditions. Caterpillar, it is a kind of larva, immature stage of a butterfly or moth. Caterpillar are the kind of larvae which are found in order Lepidoptera. Companion planting or companion cropping, planting certain plant species in close association with other crop plants as an effort to repel pest. Biotic disease, this is already covered. Now, disease, an unhealthy condition that caused by a pathogenic bacteria, fungus or virus that impairs the function or performance of an organism. Disease means dis means not, ease means easy, easiness. If something or some individual or some plant is not at ease, it is finding its life running difficult, then we can broadly call it disease. Economic threshold, the point at which damage caused by a pest exceeds cost of taking management action to reduce pest abundance or subsequent damage. Means if the level of pest has crossed the economic threshold, then there is need to apply insecticide, chemicals or other measures to control this particular problem. Exclusion. Exclusion is the act of keeping pests away from crops, structures or other areas, commonly by using barriers such as fencing, row covers or screen, etc. Fungus, a multicellular organism, for example mildews, molds, rust, smut, etc., that lack chlorophyll and derives its nutrient from other organism. Larva, the immature form of an insect that hatches from an egg, feeds and then enters a pupal stage. Pustule, a small blister-like elevation of epidermis from which spores emerge. Sanitation, activity that reduces spread of pathogen inoculum, especially the removal and destruction of infected plant root parts and cleaning of equipment and tools. Trap crop, crop or portion of a crop intended to attract pests so they can be destroyed by treating or relatively small area or by destroying trap crop and pest together. So trap crops are in fact used to attract the insect and get the main crop saved from that particular insect or that particular problem. Now we go to the insect pest details of the groundnut. So first one major insect is are aphids, jassids and thrips. So these three are very important for groundnut and lot of damage can be caused by these insect pests under certain climatic and soil and crop conditions. So these are major sucking pests. Sucking pest here means they suck the juice, they suck the sap from the leaves, maybe from the other plant parts like stem or maybe from flowers or pods also. Aphid, the scientific name is Apis gossypi. Adults and nymphs congregate mostly on growing tips and young foliage, which result in chlorotic patches and curling of leaves. As flowering commences, aphid infest flower, leaves and pegs and thereby adversely affect the pod formation. So if your sap is sucked by them, lot of minerals, lot of nutrients and proteinous matter are extracted or absorbed by these insects and plants become weaker. Large jassid population are often seen during August and September and February and March in groundnut growing areas in India. What they do, these jassids, they excrete a sticky fluid, that is honeydew, on the plant which is turned black by a fungus thereafter. So it can be harmful because the leaves or plant parts can become blackish in color and then the photosynthesis can be affected. Adult and nymph congregate mostly on growing tips in case of jassid also and young foliage and suck sap, which result in yellowing of leaves. Thrips are small insects that live in the flowers and folded leaflets of the groundnut. Nymph and adults thrips suck sap from the surface of the leaflets, initially resulting in white patches. You can see some white patches on the upper and necrotic patches. Necrotic patches here means the dead tissues on the lower surface of the leaves. Thrips, scientific name is Thrips tabaci linde. Thrips are common, dangerous to crop as they transmit peanut bud necrosis virus. So they also transmit the virus which can cause widespread plant death. Both aphids and thrips are capable of transmitting rosette disease of groundnut. So you can easily see that they are damaging the groundnut crop severely and not only damage but they can also transmit certain viruses. They spread the disease in the groundnut crop. So they are harmful in two ways. For control of sucking insect pests that is aphid, jassid and thrips, spray dimethoate 2 ml per liter or imidacloprid 0.4 ml per liter at the rate of 500 liter per hectare. Means either of these two can be chosen and mix it in the 500 liter of water and go for spraying. For thrips, insecticide application does not help much to reduce levels of bud necrosis disease. Planting of castor palm millet and pigeon pea as a trap crop is useful for controlling many of such insect pests. Intercropping with palm millet generally reduces the population of jacids. So, wherever we have severe infestation of these pests, then better to go for crop rotations and also for intercropping systems. Number 2 major insect pest of groundnut crop is leaf miner. So, you can see newly has larva of insect mine the leaves. So, in this case the damaging stage is larva. If a mine is opened, the minute caterpillar can be seen inside. You can see the caterpillar on the right side. This is caterpillar of leaf miner causing major damage to the crop. Later larvae bite the leaves and come out, web several leaflets together and make a chamber. So, that way it can be very damaging. On the right side you can see the webbing done by leaf miner on the leaf. So, due to this webbing and web formation, huge losses in pod yield are observed. A severely attacked field look like burnt from the distance. You can see it from far away that something bad has happened with this groundnut crop. For control of leaf miner, you can grow tolerant varieties like ICGV 86031. There are number of varieties, only one is listed here that can be chosen in case if you fear that this infestation will be more in a particular area. Now, coming to chemical control, it is recommended if 5 or more active larvae per plant are found. So, this is economic threshold of this insect. 5 or more active larvae per plant are found up to 30 days after emergence of the crop, 10 larvae per plant at 50 days after emergence, or 15 larvae per plant at 75 days after emergence or later. So, these are the economic threshold of this insect at different growing stage of the crop. Then what can be done is spraying of quinol phos 2 ml per liter of water at the rate of 400 or 500 liter solution per hectare should be applied if groundnut leaf miner are observed over the threshold level. What more can be done? The setting up of light traps for destroying the moth may also control this leaf miner. Rotation of groundnut with non-leguminous crops should be followed to reduce the pest incidence. Similarly, rotation of groundnut with soybean should be avoided. Collection and destruction of moth by setting light traps early in the season is advised for effective management of the pest. Now, you would surprise that damage is being done by the caterpillar or larva, but how come this management or control of moth would be useful in managing the disease? In fact, what happens if we kill the adults, particularly female, then egg laying will be stopped and then there will not be formation of caterpillars. So, better one can also control the moth, the adult insect. Now, third most important insect pest of groundnut is white grub, Holotrichia consanguinea blanche. This is a major pest of light soils, mostly in light soils and in the states of Rajasthan, Uttar Pradesh, Haryana, Gujarat and Punjab. The C-shaped white robust grubs, the shape of the grub is white, the C-shape, and they are white in color and their head or nose is orange in color and eat the plant roots and nodules, and consequently the plants will wilt and die. So, from the above you will not see any insect problem, but it is living in the soil, eating the roots and nodules of the groundnut crop, and ultimately plant will look dry because it can disconnect the roots. Severely infested fields have large patches of dead plants. The surviving plants are often stunted and slow signs of wilting. Now, you can see the grub C-shaped, the shape is C, you can see, and the head part is orange, rest is white. This particular pest is predominant in Rajasthan, Gujarat, Haryana, Punjab, even in Bihar and Uttar Pradesh, whereas H. serrata is most destructive in the state of Karnataka, Andhra Pradesh, Tamil Nadu and Maharashtra. That means it has got two different species. One is common in certain state and other is common in other states. Therefore, a holistic community approach should be adopted for the effective management of white grub. It is not that only one farmer, if he controls the white grub, the problem is sorted out. That is not the case. It has to be done on a large scale by community efforts. Now, how to control the adults? Similarly, here also, as in case of leaf miner, we need to control the adult. If adults are killed, if their population is reduced, then their reproduction will also reduce. That means production of eggs can be limited. So, how to control those adults? Trimming off trees and shrubs. So, what happens? These adults would like to stay. They would like to stay on the trees or bushes nearby area to the field. So, let us clean those kind of shrubs and bushes, placing 3 pheromone traps, synthetic pheromone anisole dispensers per tree at 15-meter radius for 3 consecutive evenings after the first monsoon rains. So, after first monsoon rain, this pupa can hatch into adults, and at that time these kind of measures can be adopted, like putting the pheromone dispensers. Placing light traps at the rate of 1 trap per hectare between 7 p.m. to 10 p.m. because they come in the night after receipt of first monsoon rains. Then spraying insecticides such as imidacloprid 17.8 SL at the rate of 1.5 ml per liter during the evening hours on trees and shrubs, and collecting and destroying the adults fallen near the base of trees, it is recommended. So, this is how we can control or we can reduce the population of the adults. Now, control of grubs which is actually damaging stage. So, deep summer plowing to expose the pupae to scorching solar radiation and predation by birds. So, this is a good option, deep summer plowing. So, it not only controls these grubs, but it will control other insect pests also. Even weed seeds can be killed by deep summer plowing. Crop rotation with jowar or sorghum and bajra or pulse millet, and incorporation of carbofuran 3G at the rate of 33 kg per hectare in the soil before sowing. If you assume that you have lot of white grubs in your soil, then you can go for application of carbofuran 3G at the rate of 33 kg per hectare. Furrow application of thiamethoxam. Other option is thiamethoxam 25 Ws at the rate of 1.9 liter per hectare or fipronil 5-FS at the rate of 2 liter per hectare. These can also be applied. Drenching root zone with chlorpyrifos 20 EC at the rate of 4 liter per hectare or quinalfos 25 EC at the rate of 3.2 liter per hectare 3 weeks after emergence of adults. So, you need not to follow all these chemical application. You can choose either of these, any one of these. So, now the fourth one, insects are actually caterpillars, and there are number of caterpillars that affect this crop. So, there can be red hairy caterpillars, Insecta albistiga, tobacco caterpillars, Spodoptera litura, very damaging insect, and gram caterpillars, Heliothes armigera, are chewing and defoliating types of insects that destroy the entire foliage, leaving the midrib. So, most of these caterpillars, they eat the leaf or leaves of the groundnut plants. For control of Spodoptera litura, monitoring the pest from September last onward by pheromone traps. So, monitoring is necessary for this litura. Spraying neem oil 5 ml or chlorpyrifos 2.5 ml per liter of water at early stages. Spraying of NPV, nuclear polyhedrosis virus 500 LE per hectare. This is the dose 500 LE per hectare from third instar larvae and spraying of thiodicarb 1 gram per liter of water or for late instar larvae are recommended. So, you have options of chemical control and option of biological control in the form of NPV to control the Spodoptera luteura. And then for control of red hairy caterpillar, use of trap crops like cowpea around the main crop, monitoring the emergence of adult moth through the light trap, dusting with quinol fas at the rate of 25 kg per hectare to control early instar of the caterpillar, and to control grown up larvae, spraying of dimethoate at the rate of 2 ml of water is recommended. This dimethoate is actually Rogor. The rate of application is 2 ml per liter of water, and one can make about 500 liter solution and spray in 1 hectare area. For now overall, what can we see? The improved plant protection measures for insects. Overall, you can see for managing thrips and leaf hoppers. These thrips and leaf hoppers are usually suck the sap of the plant. So, what can be done? A single spray of imidacloprid 20 SL at the rate of 150 ml per hectare, or another option is thiacloprid 480 SC at the rate of 125 ml per hectare, or thiamethoxam 25 W G at the rate of 100 gram per hectare, or acetamiprid 20 SP at the rate of 100 gram per hectare between 25 and 30 days after sowing or on need basis. But here we should remember that spraying of these chemicals should be done only the insect has crossed the economic threshold level. For managing the defoliator pest like Spodoptera luteura and Helicoverpa armigera, a single spray of Rynaxypyr 20 SC at the rate of 25 ml per hectare, or flubendiamide 480 SC at the rate of 100 ml per hectare on need basis. So, these are the recommended insecticide. Either one of these can be used. For managing leaf miner, that is Aproprema modicella, a single spray of profenophos at the rate of 50 EC, 50 AC 1000 ml per hectare, or spinosad 45 SC at the rate of 150 ml per hectare, or quinol fas at the rate of 1000 ml per hectare on need basis. But again, if you have experienced the economic threshold level of the insect, then one can go for spraying of these insecticides. Seed treatment is also good for managing the leaf miner. Seed treatment with chlorpyrifos 20 EC at the rate of 12 ml per kg seed, or if it is not available, we can take imidacloprid 48 FS at the rate of 2 ml per kg for management of white grubs, followed by placing light traps at the rate of 1 trap per hectare between 7 pm to 10 pm after receipt of first monsoon rains. Spraying of imidacloprid 17.8 SL at the rate of 1.5 ml per liter in evening on tree shrubs for white grubs. So, this is how you can control the adult of this white grub as well as the grubs, the caterpillar in the soil. Now, just now you have seen that we should go for insect pest management because they cause lot of damage to the crop. They can destroy the foliage part, leaf part, and they can suck the sap also. They can reduce the yield of the pods, the quality of the pods or quality of the seeds. So, overall we are going to have losses from the insects, and the best way to manage the insect pest is through integrated pest management strategies. Integrated pest management strategies include even time of sowing. By adjustment in time of sowing, we can reduce the population of certain insect pests. Also, there is necessity to control the vectors. Sometimes insect can carry certain viruses, so those vectors can be killed. Also, it is necessary to control some adults moth that can lay eggs and which can convert into larvae after the hatching. So, control of adults is also required. So, we can go for crop rotations also. We need to rotate the crop with other crops, this groundnut crop, and also proper plant population. Seed treatment is also a good option because here the investment is less, but the results are good. So, seed treatment can be adopted, light trap can be used, pheromone traps can also be used. Even certain biological agents are available like NPV to control the insect pests and diseases. So, better to go for integrated way, and chemical application or application of herbicide should be in minimum doses, and need based application can be done. So, what this IPM is going to do, there will be profitability, means profitability of the cultivation of groundnut will improve, and also the environment will be protected. Less application of insecticide into the soil means good soil health, and also less application of insecticide to plants means also good human health. Because if you apply excess doses of pesticide, they can reach to the food chain, they can come to our table. So, therefore, from all perspective, integrated pest management is the best way to control, to manage the insects. So, after insect pests, now let us go to the disease management, which is equally important, and we will take one by one these diseases. So, early leaf spot is number one disease. It is caused by Cercospora arachidicola, Cercospora arachidicola, or it is also called as ticca disease of groundnut. It appears on lower leaves during 10 to 30 days after emergence. Appearance of circular spot like this, you can find circular spots that are brown to reddish brown on the upper surface of the leaflet and lighter shade of brown on the lower surface, surrounded by yellow halo. Temperature between 25 to 30 degrees centigrade and high humidity, high relative humidity more than 60% are favorable for incidence of the disease. So, you can see the leaves or leaflets of groundnut here. These both are symptoms of early leaf spot or ticca disease. You can see in the middle they are brownish spots, and on their periphery you can see yellowish color, and then these spots can increase in size also, and they can cover the whole leaf, and then the photosynthesis will be stopped, and there may be problem with the growth of the crop. So, and so far control is concerned, we will discuss it later. Now, you see the late leaf spot. Late leaf spot, it is caused by P. personata, dark brown to black circular spots, almost same as in case of early leaf spot, but here you get dark brown or to black circular spots on lower leaflet surface, which are darker than the early leaf spot. This is the only difference. They are darker than the early leaf spot and have a feathery margin. In case of severe incidence, affected leaflets become chlorotic, then necrotic, followed by coalescence of the lesions, resulting in premature senescence and shedding of leaflets. So, this is quite interesting, and stage by stage you can remember the symptoms. First, the leaf will become chlorotic, means there will be loss of chlorophyll or yellowing of leaves, then necrotic, then those area will die, and followed by coalescence, means they will merge into each other of the lesions and resulting in premature senescence, means maturity, and shedding of the leaflets. Now, you can see dark brown spots, very dark or blackish spots. So, these are the typical symptoms of late leaf spot, and you can see the leaf, this particular leaf is drying from the tip, and you can see the differences in late leaf spot and early leaf spot. On the left side you have early leaf spot, you can see these reddish brown spots here. The spots are reddish brown, and on the right side they are blackish or dark brown in late leaf spot. This is very minor difference between these two, and people get confused about early leaf spot and late leaf spot. Although the causal organisms are different. So, prolonged wet weather, high humidity and high temperature, same as it was for early leaf spot, are favorable for the incidence of late leaf spots. Yield loss can be as high as 50% in case of severe infection affecting pod filling and maturity. Now, management of both early and late leaf spot together. What can be done here? A serial groundnut crop rotation should be followed. Means there should not be legume rotation. Deep plowing should be done during land preparation, because this deep plowing, what it will do, it can be done in the month of say late April or early May, and then it will expose to this fungus and all kind of disease causing organism that can be killed by the scorching sun. Infected crop residues should be removed and destroyed. Field sanitation, cleaning of bunds, all these measures should be adopted, and volunteer groundnut plants should also be eliminated. So, these measures are necessary, and then sowing dates should be adjusted so that the most conducive environment for disease development can be avoided. And then foliar application of carbendazim. Carbendazim is also known as bavastine. The concentration is 0.05% plus mancozeb 0.2% at 2 to 3 weeks interval. So, you need 2 to 3 times spraying of these fungicides together, starting from the initiation of the disease. When you think that the disease has been initiated, then you can go for spraying of these 2 fungicides in combination. Cultivars tolerant to early or late leaf spot like ICGV 87160 or Somnath. There are some other cultivars of varieties which are tolerant or resistant to these 2 fungal diseases. Those may be grown in area where the disease has become endemic. Now, third important disease of groundnut is rust. So, it is caused by Puccinia arachidis. Puccinia arachidis. Appearance of numerous tiny reddish orange pustules on the lower surface of leaflets. It is starting on the lower surface of the leaflets initially and on the upper surface at a later stage. So, beginning happens from the lower surface, but in advanced stage these pustules can come to the upper surface also. Through it appears predominantly on the leaf surface. In case of severe incidence, the rust cover all the entire aerial part of the plant. Means this rust can spread to the stem, to the branches, to the buds or to the flowers or maybe to the pods also. As a result of the infection, the leaves become necrotic. Necrotic means death, and dry up and have a scorched appearance affecting pod maturity and yield. So, you can see the symptoms here, very small tiny spot or pustules of reddish brown color on the leaves. So, these are typical symptoms of the rust on groundnut crop. So, what can be done for management of rust? Resistant and tolerant varieties can be grown like ICGV 87160, ICGV 86590, ICGV 86325, TAG 24, ALR 3, VRI 5, CSMG 84-1, ICGS 5, GJG 32, KDG 128, KDG 123, etc. can be grown, and removal of infected leaves will spread the secondary infection to the healthy leaves, and this clearing of branches or diseased branches will definitely help in reducing the spread of this disease. Seed treatment with tabuconazole 2 ds at the rate of 1 gram or 3 gram mancozeb per kg of seed. So, seed treatment is also useful. Spraying of mancozeb 1000 gram or chlorothalonil thalonil at the rate of 1000 gram or 1 kg dissolved in 500 liter of water at 15 days interval starting from disease appearance. So, many times fungicides like mancozeb or maybe chlorothalonil or maybe bavestine spraying can be useful to prevent this disease. Now, fourth important disease is collar rot. The causal organism is Aspergillus niger, infecting collar region of the seedling. This collar region of the seedling is mainly the part of the plant where the stem and roots join together or they meet together. Infected areas are covered with black fungal spores. In extreme severity, the whole plant wilts and wilts permanently. Wilting means drying of the plants. Areas in Rajasthan, Gujarat, Punjab are badly affected by the incidence of collar rot with seedling mortality 25 to 50%. Means it is more dangerous in early stages. If your seedlings are killed, if seedlings are destroyed, then definitely you will not have the optimum plant population, which will adversely affect the productivity of the groundnut crop. The spores or hyphae of fungi present in the soil or adherent to the seed surface means it can be soil borne and it can be seed borne. They germinate and attack the seed, causing pre-emergent rotting of the seed. That can be quite dangerous. The other characteristic diagnostic symptoms are rotting of hypocotyle. Hypocotyle is the region of the seedling below the cotyledons. Post-emergent seedling blight. Even after emergence, blighting of the seedlings can happen, and rapid wilting of the entire plant or its branches. So, many times it can be quite harmful disease. The collar region of the affected plant becomes shaded. Shaded here means split, kind of split, and becomes dark brown to black, covered by mycelia and spores, hence the name collar rot. Now, you can see this region of the plant, it has rot. So, then the plant will start wilting. So, you can see early symptoms of the collar rot on the left side, and on the right side drying up of the seedling. At early stage the seedlings are drying. So, this is really can be very dangerous. Now, what should be the management options for collar rot? Deep summer plowing with mold board plow followed by seed treatment. Seed treatment can be done with tabuconazole or therum at the rate of 1 gram per kg seed, sowing at 2.5 centimeter depth, and soil application of trichoderma at 4 kg per hectare enriched in 50 kg farmyard manure are recommended for effective management. So, here you can say you can use the chemical seed treatment with tabuconazole or therum. In addition to chemical seed treatment, one can also apply this trichoderma directly to the soil to manage the soil-borne fungus or seed-borne fungus. So, trichoderma at 4 kg of trichoderma can be mixed with 50 kg farmyard manure, and it should be uniformly applied to the soil before sowing. Now, fifth disease is stem rot. Causal organism is Sclerotium rolfsi, infecting stem just above the soil surface or at the foot of the plant. So, you can see name comes stem rot, rotting of the stem. Initially, deep brown lesions appear around the main stem at the soil level. The distinct rot occurs beneath the fungal weft, leading to wilt-like symptoms characterized by yellowing and browning of the foliage, which shows drooping while remaining attached to the plant. Here drooping means hanging. Rotting of seed before germination if seed comes in contact with germinating sclerotia. Sclerotia are the propagating unit for the fungi, which can remain dormant, and under ideal conditions they can give rise to new growth of the fungus. Rotting of cotyledon and young seedlings if germinating seeds come in contact with growing mycelia of the fungus. So, you can see that the Soil-borne fungus is also dangerous. The initial symptoms are partial or complete wilting of the stem or branches that are in contact with infected soil after generally 30 to 40 days after sowing. So, you can see it is primarily a soil-borne disease. If we go for adoption of crop rotation, we can surely reduce the fungus growth, and if we take cereal crops or sugarcane crops in the rotation, that will surely reduce the population of the fungus in the soil. White mycelium is visible on the infected plant parts. On the right side, you can see the stem rot. The rotting is in this part, lower part of the stem, and you can see the white growth of the fungus in this picture. The leaves turn brown and show wilting but remain attached to the plant and eventually drying up plants with the rotten pods. Yield loss can be as high as 60% due to incidence of the stem rot. Now, management of stem rot, deep summer plowing, as has been the case for other fungal borne diseases also. So, deep summer plowing with moldboard plow followed by seed treatment with tabuconazole or therum at the rate of 1.5 gram per kg of seed, and soil application of trichoderma at 4 kg per hectare mixed in 50 kg farmyard manure, and it should be uniformly applied to the soil in 1 hectare area, and application have to be uniform and before sowing. It should be mixed in the soil also. Now, sixth one is charcoal or dry root rot. This is also important disease caused by Macrophomina fasciolana. It is the causal agent of the disease. Appearance of water-soaked lesions in the hypocotyle region of the young plants near the soil surface. In older plants, the infection starts near the soil surface and spreads to the stem and branches and downwards to the roots. So, it can affect many parts of the plant system. Sometimes the disease may be restricted to roots, which become brown and on infection and lateral roots become black and rotten. Seed treatment can be done with Trichoderma viridis at the rate of 4 gram per kg seed, or seed treatment can also be done with carbendazim at the rate of 2 gram per kg seed, or therum at the rate of 3 gram per kg of seed recommended for the management of this disease. Now, next is peanut bud and stem necrosis diseases, PBND and PSND. So, in PBND is peanut bud necrosis disease, is caused by peanut viruses, necrosis of the terminal bud. A characteristic symptom occurs on crop grown in the rainy and post-rainy seasons. The secondary symptoms are stunting, auxiliary shoot proliferation, and malformation of leaflets. Seeds from early infected plants are small, shriveled, mottled, and discolored. Seeds from early infected plants are small, shriveled, mottled, and discolored. In PSND, the entire plant is often subjected to necrosis if the disease appears at an early stage. In the case of older plants, one or more branches will show necrosis symptoms. However, other branches look healthy. These plants are stunted. Stunted means their height is reduced or they are dwarf and do not show any axillary shoot proliferation, means lateral branching may not be there. Necrotic spots are observed on the majority of pods. The size of the pod is adversely reduced and kernels are not marketable. So, you can see sometimes there can be huge loss to the quality of the crop as well as to the quantity of the crop. Bud necrotic disease is transmitted by thrips, which can be controlled by spraying Rogor at the rate of 400 ml per hectare. You can use this Rogor or dimethoate. So, this disease is killing the thrips. This particular insecticide will kill the thrips, which will not be able to transmit this disease from the diseased plant to the healthy plants. The culture methods such as early planting, closer spacing and intercropping with millets are advocated or suggested to manage this disease. Now, the last one is nematode diseases. So, nematode are very small organisms which are not seen by our naked eyes, and they live in soil, they live in plants, and sometimes they can also cause diseases in crops. And there are cases when nematode infestation also happens in case of groundnut crop. So, it has been reported from Punjab and Gujarat that the nematode Melardogyni arenaria infestation causes a significant reduction in the growth and yield of groundnut crop. The attacked plants remain stunted, their growth is reduced, and chlorotic means greenish or yellowish, and the roots get profusely galled with lateral roots. The galling means kind of knots will be formed on the roots. The presence of May arenaria increases invasion of shell and kernels by Aspergillus flavus. So, they will be further attacked by the fungus Aspergillus flavus, which can produce aflatoxins also. We will discuss in detail about the aflatoxins. On the roots and pods, galls are formed. Galls are actually kind of swelling. Galls are formed associated with lateral roots, giving a matted appearance to the root system. Another nematode, A. arachaitis, lives endoparasitically in testa, causing seed coat distortion. So, it may be at fruiting stage or at pod development stage. The testa is wrinkled and becomes dark brown with a small shriveled seed which gets predisposed to fungal infection. So, that is the problem. Is first thing is that nematode itself is doing the damage, and secondly this damaged part will invite the growth of the fungus. So, it is double whammy for the plant. Pratylancus species, which is known as the lesion nematode, causes dark color lesions on the shells. Shells are the kind of coat of the fruit, that is pod. The shells serve as the source of the initial inoculum. Inoculum is disease causing part. It is also associated with rotting of the pegs, which are important for fruit development in groundnut. Besides this, it causes chlorosis and stunting of the plant. Now, what are the management options for nematode diseases? Deep plowing, rotation with cotton and flooding also reduces nematode infestation. Application of entomopathogenic nematodes is also recommended for the management of nematodes. Some options are available where nematode can be used to control the nematode. That is also again a kind of biocontrol method of management of nematodes. Up to now, you have seen the insect pest and disease management in case of in groundnut. Let us see harvesting and threshing. Now, your crop is at physiological maturity or harvest maturity, and you are going to harvest. How it is done, let us know. The harvesting depends upon the type of variety grown, season and management practices. Early as well as late harvesting of the crops affect the yield and quality of the produce. So, crop should be harvested at right time. The prominent symptoms of maturity are the yellowing of leaves, shedding of older leaves, development of the proper color of the testa and dark tint inside the shell. So, these are the symptoms or observations one can take to consider the harvesting stage of the crop. Usually crop takes 110 to 140 days to mature, and the harvesting is done in October to November. Of course, the maturity period will be determined by the variety. We have different varieties of different duration. In case of bunch types, the plants are harvested by pulling. So, harvesting of bunch type groundnut is quite easy. You can harvest it simply by hands pulling it, whereas in spreading type, harvesting is done by spade or sickles or some other implements, country plow or groundnut lifters. So, lot of options are there. Some new machines have come up because in spreading type the roots are deeper, much deeper and much wide. In case of bunchy type, root growth is less. Therefore, it is easier for people to uproot the bunch type groundnut, but in spreading type you can use spade or some other implements. The harvested crop is left in small heaps for 2 to 3 days for curing or kind of drying. After curing, collect the crop at one place and detach the pods either by hand or using a groundnut plucker for separating the pods from the plants. So, separation of pod is required. By adopting improved technology, it is possible to obtain 1.5 to 2 tons of pods per hectare. This is the yield from bunchy type, and for spreading type the yield is 2 to 3 tons per hectare. So, you can see the spreading type varieties of groundnut give you more yield than bunchy type. However, these spreading type are late maturing, mostly late maturing compared to bunch types. Average shelling percentage is 70%. This shelling percentage means the percentage of the seed in the fruit or in the pods. What proportion of seed is available from the pods? Oil to nut in shell is 28%. Oil to kernels crushed is 40%. This value student can remember. Oil to kernel crushed is 40% oil and cake to kernels crushed is 60%. Naturally, if 40% is oil, then 60% can be the cake, and the oilseed cake of this groundnut is very valuable. It can be used as animal feed, and it also finds some other uses, but most prominent use of groundnut cake or seed meal is for animal feeding. But at the same time, one major problem encountered by groundnut is of aflatoxin. Sometimes the fruits and seeds of groundnut get infested or get attacked by aflatoxin or get contaminated by aflatoxin. So, let us see more about aflatoxin. Aflatoxin contamination is a serious problem in most groundnut producing countries. Aflatoxins are produced. So, these are actually organic chemicals or poisons that are produced by toxigenic strains of fungi like Aspergillus flavus, Aspergillus nomeus and Aspergillus parasiticus before harvest. So, they can come before harvest and even after harvest to the seed or to the pods, during post-harvest curing and drying or in storage. So, after fruit development or development of pod, these kind of fungus infection can occur at any stage, before harvest or after harvest or in the storage. Contamination is largely a pre-harvest phenomena in the semi-arid tropics. Most of the time it is pre-harvest, but that does not mean that it is always pre-harvest. It can also happen post-harvest. So, aflatoxin contamination management, how to manage it? Aflatoxins are highly toxic metabolites, these metabolites being carcinogenic. That is why there is hue and cry around these metabolites, mutagenic and immune suppressive, pose serious health hazards to humans and animals. Of course, I am not going into the much details of this subject, but overall you can see that it is not good for human and animal health. We should not have aflatoxin in groundnut. Now, pre-harvest aflatoxin contamination. In light arid soils or you can say sandy soils and alphy soils favor rapid proliferation of these fungi, and risk of pre-harvest aflatoxin contamination are high in these soils. So, there appears to be less risk of pre-harvest contamination of groundnut in vertizoles. So, these kind of contaminations can come from soil, they can come from seed, etc. Now, what can be done to stop or to manage the pre-harvest aflatoxin contamination? Deep-plowing 8 to 10 inches in summer. The main purpose here is to suppress the fungus so that infection does not occur. Apply well-decomposed farmyard manure at 5 to 10 tons per hectare. Apply neem cake at 500 kg per hectare at the time of sowing to control the fungus. Select short-term or medium-term groundnut varieties. Seed treatment is also good with tebuconazole 2-DS Rexil at the rate of 1.5 gram, or seed treatment can also be done with carbendazim 50 WP at 2 gram per kg seed. Advance sowing by a fortnight by 15 days. Follow inter-row water harvesting by adopting pair row method of planting. Avoid end of season drought by providing supplemental irrigation for rain-fed groundnut, and apply gypsum at the rate of 400 to 500 kg per hectare, because gypsum contains sulfur and that can suppress fungus. Post-harvest aflatoxin contamination. After harvest, mechanical damage to the pods or kernel at harvest or threshing, and decortications, and inefficient and slow drying and storage of produce in warm and humid rooms favors aflatoxin contamination. So, these are the conditions or mechanism by which post-harvest contamination can happen. So, how to manage this post-harvest aflatoxin contamination? Harvest at right time when the crop is fully mature. Avoid mechanical injuries to the pods. Do not mix immature and leftover pods with the harvested mature pods. Mixing can also bring contamination. Dry the pods to a safe moisture level of 8%. Defective pods or kernels should be separated. Proper drying, proper shelling, post-harvest processing. Technologies such as sorting of peanuts and camera or laser sorter and by hand picking are likely to remove aflatoxin contamination kernel. Now, machines are available where we can separate these infected kernels also. Finally, see the seed oil and cake quality. 4 types of groundnut under cultivation. They are Virginia runner, Virginia bunch, Valencia and Spanish bunch. They differ with respect to their chemical composition and oil quality. For example, Virginia bunch type group is richest in oil and energy content, followed by Spanish bunch. Protein content is higher among Valencias, while soluble sugars are higher in Virginia runner. With respect to oil keeping quality, that is oleic, linoleic ratio, runner type is better, while Valencias possess a higher nutritional quality index. Hence, Virginia runners have been considered to be better with respect to their quality. This point must be underlined by the students. Summer groundnut is good for higher oil yield and better oil quality, but lower levels of sugar and proteins. So, in all these groups of groundnut, some have positive properties and some do not have that much positive properties. So, one can choose according to the requirement. Cake and meal is also second major product of the oilseed processing industry. Lower the fiber, the higher the protein content of the final feed and the more attractive it is. The cake or meal has a small quantity of oil, 0.5 to 1%, trace elements and impurities. Therefore, cake can be used as animal feed. Dear student, hope you enjoyed this lecture. Thank you very much.